So we're going to talk through the problem that is included as the mammoth gene question and kind of the motivation behind it, and then also how to solve the problem. So within this topic of de-extinction, genetic engineering is considered by many to be the most promising pathway to actually achieve this goal of de-extinction for the largest number of species. This is because the other pathways that are available are really limited to which species they can be used for because of what they need in order to be successful. And with the technological advancements in genetic engineering and cloning technology in like recent years, it's becoming more and more likely that genetic engineering is like the best pathway for the most species to actually achieve this de-extinction goal. And so to do this, genetic engineering, to edit those specific genes that are going to help transform this extant species that you have into having the traits and characteristics of the extinct species that you want it to. Geneticists once would have had to do this by hand, editing each of these genes one by one to change one trait for the other. And so this is kind of what we're exploring with this question here. But today, in actuality, this process has been automated in many ways. So that not as much of it has to be done by the geneticists by hand, but understanding kind of this basic science behind it and this basic genetics is really going to help us not only to understand those more modern science applications, but also to keep in mind as we're having these discussions on like the ethics and the social implications and the policy and legal questions that we kind of pose throughout this website. And so, what this problem is really asking is for you to consider a scenario where you've already cut out a gene from the woolly mammoth's genome. And this gene that you've cut out is going to code for cold adapted hemoglobin, which is what allows your mammoth to, which is what allows the woolly mammoth to efficiently transport oxygen in its blood, even at cold temperatures. And so, this is something that your mammothant, resurrected species is definitely going to need if it's going to survive in those cold Arctic temperatures. And so now that you have this sequence cut out with these sticky ends that in the middle somewhere contains that gene for your cold adapted hemoglobin, you need to insert this into your elephant DNA. You need to replace the elephant's current hemoglobin coding gene, which wouldn't be able to make cold adapted hemoglobin, with this one from the mammoth that can, because your mammothant combination needs to be able to have that cold adaptation for their hemoglobin. And so, in order to do so, this problem is going to ask you to consider using a restriction enzyme to cut that elephant DNA on either side of this hemoglobin gene, and then to insert in the mammoth gene. And so, the way this problem is set up, it's broken down into four parts to kind of help guide you through what you need to think about and through the basic steps of this process so we can get a better understanding of it. So the first question here says to identify the restriction sites in the elephant DNA that have palindromic sequences. So palindromic sequences are going to be inverted repeats within your DNA, which are complementary to themselves. So in fact, the DNA could fold and pair to itself instead of having to pair to that other strand in this double-stranded formation. And so we're going to highlight these here in pink just so that they're nice and easy for us to see. And so the first one is going to be right here, this AAT GC GCATT, and the complementary strand here. So you can see that if this were to fold in half, the C compared to the G to the C, the T to the A to the T, and the A to the T. And so then we see this same sequence again over here. And so both of these palindromic sequences are going to be those recognition sites for the restriction enzyme that we're using so that it can figure out where in this very long strand of DNA, to which this is only like an excerpt, it's going to be making those cuts. So the next part of the question gets us talking about that restriction enzyme. So if you're using a restriction enzyme that cuts two nucleotides from the axis of symmetry in these restriction sites, creating 3' sticky ends, identify the axis of symmetry within each palindromic sequence and where the cuts will be made. So some important things we want to pull out of this question is that we have a restriction enzyme which cuts two nucleotides from the axis of symmetry, and that it creates 3' sticky ends. So those are things that we want to keep in mind as we're going through this process. But the first thing that it asks us is is to identify the axis of symmetry, which we're going to do so in blue here. So we can find that, like we were discussing before, is the point within this palindromic sequence where it could actually fold on top of it, on half or on itself. And so what is that kind of like middle point where that folding could happen? And so it's going to be right between this C and G, and then the same over here. And then I'll drag out this label just so that we can remember that our blue lines are showing us our axis of symmetry. So the next part of this question is asking us to show where the cuts will be made. And so that we're going to do with this green color. And so it told us before, remember, that it cuts 2 nucleotides from the axis of symmetry. So we're moving 2 nucleotides away from this center line that we identified already. And then it's creating 3' sticky ends. So when a restriction enzyme creates 3' sticky ends, it means that it's going to be moving 2 nucleotides towards the 3' end of the DNA before it makes that cut. So here's our axis of symmetry. So we're going to move 1 nucleotide over, 2 nucleotides over, and then make a cut through that first strand. Then we're going to cut through these hydrogen bonds between the strands, and then 2 nucleotides towards the 3' end on the bottom. We're going to make that cut again. And then we see the same thing over here. Again, 2 nucleotides towards the 3' end, make that first cut, cut sideways through those hydrogen bonds, and then we're going to cut through that second strand, again, 2 nucleotides towards the 3' end. And so we have a nice label here too to show us that our restriction enzyme cuts are in green, just so that we remember as we move on kind of what those different lines meant. So now that we know where those cuts are being made and where that axis of symmetry is, we can think about how those cuts would actually look. And so bringing up this representation, this is the same sequences of DNA that are included here, just including space so that we can better visualize those cuts. So if we add back in where those cuts were made, it would have been right there, right here, right through that space on either side. And so then if we think about, again, where our palindromic sequences are, just so that we don't lose track of them, that's these, and then here, and these, and there, and then the same thing over on this side again. And just so that we can drill it into our brains, we'll also draw in our axis of symmetry again. And so it's not going to line up perfectly because we have these spaces added in, but each of these blue lines shows where it is in the individual strands. And so now that we have this piece in the middle, this is going to be the sequence that's going to have included our hemoglobin gene. And so we're going to get rid of that and switch in our mammoth, our like hemoglobin gene from our mammoth, and see if it can like pair with our sticky ends that we created. So now bringing up this one, you'll notice that this center section has a different sequence. This is because this is that sequence that we already had isolated from our mammoth gene. And so now we can look and see if our mammoth gene is going to be complementary to our elephant sticky ends that we created. So what we really need to look for is where there would be those overlaps. And so you can see right here, it's going to be this GCGC, that's like sticky end that we'd created, is going to pair up with the CGCG in the mammoth gene. And then the GCGC from the mammoth gene pairs up with that sticky end CGCG from our elephant gene. And so or elephant DNA. So it's showing you that we do have, like, we've used the same restriction enzyme for this mammoth gene to cut it out, and then that we just used on our elephant DNA. So it produced sticky ends that were complementary to each other, which makes it really easy to switch that in. And so then this third question, which we're already kind of starting to answer, is showing how the mammoth gene could be inserted into the elephant DNA after it's been processed with this restriction enzyme. So we already started showing it, but then to help us really visualize that, we can bring up this last representation, which gets rid of any of those spaces that we'd added in, but we can show again where those complementary sequences were. Actually, first, I'm going to highlight in yellow just where that mammoth gene is, so that we can keep it separate from where our elephant gene would have been. And that's going to be right here. And so then when we're considering looking at those complementary sequences, so the same ones that we had in purple up here will show in purple again. And so that's going to be this GC, GC, and then down here, this CG, CG. And the same on this other side. And so this is really showing you that we were able to just, with those sticky ends, quickly insert in the mammoth gene, the mammoth gene for that cold adapted hemoglobin, where there would have been the elephant's gene for their non-cold adapted hemoglobin. So the last part of this question says, what type of restriction enzyme was used to cut these sequences? So with what we know, we can't actually identify the identity of the restriction enzyme, but that's not really important for understanding how the process works. What this is asking us is kind of there's three main types of restriction enzymes: type 1, type 2, and type 3. So we're using a type 3 enzyme in this problem because we had it cut within those palindromic sequences. So type 1 and type 2 restriction enzymes, they recognize those palindromic sequences, but then they move a number of base pairs away that's going to make it so that the cuts they actually make are far outside of the palindromic sequence. Our restriction enzyme, though, if you remember back up in part 2, it just cuts 2 nucleotides from the axis of symmetry, so it's cutting right within that pink highlighted palindromic sequence. So I hope that this was able to help you understand both kind of the meaning behind this question, and then also walking you through kind of what it's asking and what it would be looking for in an answer. If you want, there's also a PDF uploaded that includes a written explanation for how to solve the problem that can help you walk you through all of these same steps we just went through verbally in just a written format.